Hi everyone, tonight's video is on membrane transport, and it is a follow-up video to the video on diffusion and also the video on membrane structure. So you should have watched those videos before watching this video. The question we want to ask tonight is, how do large, charged, or polar substances get across membranes? Things like salts and ions, they're charged. Sugars, they are polar. Water, very polar. Proteins, large, and they're very often polar. So how do these substances get into and out of cells? Clearly, they're not membrane soluble. They cannot pass through a cell by simple diffusion. Facilitated diffusion is how substances such as these cross the membrane using a transport protein or a transport channel. And that's diagrammed here in this picture of a membrane, where you can see a transmembrane protein that has a channel down the middle that's going to allow these large or polar or charged molecules to pass through the membrane. This process does not require energy. It's another form of passive transport. So there's two possible mechanisms for facilitated diffusion. Both of them use this transmembrane protein or a transport protein. So the first example here I'm going to show you is simply a transmembrane protein that forms a tunnel through the channel, and it's going to allow charged and polar molecules to move through the membrane from one side to the other, going from a high concentration to a low. So facilitated diffusion is still driven by a concentration gradient. It doesn't require energy, but it just requires a way for something that's polar or charged to pass through this large layer of fatty acid hydrophobic tails of the lipid. It does this by being protected by a protein channel. Another way that this can happen is a membrane protein that is a transmembrane protein can actually bind to a substance on one side of the membrane where there's a higher concentration, and then it actually lets go on the other side, therefore kind of moving it from one side to the other. Again, the protein or the substance gets to move through this transmembrane channel, kind of protecting it from these fatty acid tails. There is no energy required, but it does require a concentration gradient. So substances move from hyper to hypotonic or high concentration to low until an equilibrium is reached, just like standard diffusion. What about water? Water is very polar. So water can pass through a lipid bilayer on its own, but very slowly. Water channels, most membranes have these water channels called aquaporins, and some membranes have more of them than others, and they allow for a faster transport of water across membranes. For high school biology, you should assume that cellular membranes are slightly permeable to water, or at least have water channels, unless you are told otherwise. So what about moving substances against a concentration gradient? Both of the forms of diffusion that we have talked about in tonight's video and the previous video on simple diffusion move particles from a high concentration to a low to reach an equilibrium. So both diffusion and facilitated diffusion move from high to low concentration until an equilibrium is reached. And facilitated diffusion is shown here, where particles are moving from the high to the low concentration. That would be going from hypertonic to a hypotonic solution. Both of these forms, the one that is shown here and the simple diffusion shown in a different video on this YouTube channel, require a concentration gradient, but they do not require energy. Moving substances up a concentration gradient, so from a low concentration to a high concentration, from a hypo to a hypertonic concentration, in other words, that requires both energy and a transport protein. The process for moving things against a concentration gradient is called active transport. And this is where you go from a low concentration to a high concentration. You can think of this like pumping something from low to high. It definitely requires energy. That energy is in the form of ATP. ATP as an energy source is covered in a different video on this YouTube channel. Why would a cell want to move something uphill? Well, maybe the cell would like to pump food into the cell, or maybe pump waste out of a cell. Another thing cells use active transport for is to form a concentration gradient. So by pumping a substance like an ion into a membrane-surrounded location, it's then forcing a high concentration in that membrane location, and this can be used later. It's a form of potential energy. So when you think of active transport, I always want you to think about pumping. What about substances that are too large to go through a membrane or even go through a transport protein? How do they get into and out of a cell? Well, there's a process called endocytosis for bringing very large particles into a cell, and that's shown here, where the cell or plasma membrane actually reaches out and envelops a particle. It could be something as large as a bacteria, and it's brought inside the cell in a vacuole. And in this case, if it's a bacteria, the cell could then break down this bacteria, and our immune system does this to fight bacterial infections. This causes the cell membrane to actually shrink, because this vacuole that's formed here is from the cell membrane. So if the cell is doing constant endocytosis, its cell membrane is going to shrink, because some of that membrane is being used to form the vacuole that goes inside the cell. There's another process called exocytosis, where very large particles are brought to the cell membrane by a vesicle and released outside the cell. When this vesicle membrane fuses with the cell membrane, the particles can be released to the outside of the cell. This causes the cell membrane to grow, because in this case, the vesicle membrane is being added to the cell membrane. It's fusing with the cell membrane. So in addition to releasing particles that are too large to pass through the membrane or pass through a transport protein, the cell membrane is growing. So in order for a cell not to either get much smaller or much larger, it needs to do a balance of endocytosis and exocytosis. If it is an imbalance, the cell will either grow or shrink. Both of these processes require energy to move across the membranes and move the vesicles and move the vacuoles. It's actually a very expensive process. So you should know how polar and charged molecules get across membranes, what the requirements are for this process, and what's the driving force and the requirements again. That's repetitive, I'm sorry. You should know how cells move substances across a concentration gradient, against a concentration gradient. You should know what the driving force is and what the requirements are. And you should know why. Why would a cell want to move substances against a concentration gradient? You should also know how very large particles and molecules get into and out of cells and what's the driving force for these processes. So that's all for tonight.